Okay, so for experiment 3, we're going to be discussing the regulation of gene expression, or when is a gene turned on, and what conditions are required for the gene to be expressed. Okay, so gene expression is also when and where is the gene turned on. Okay, so there's a whole class of genes that are always on, okay, and these are called constitutive. Constitutive expression. Okay, so a constitutive gene might be like glucose 6-phosphatase that is required to utilize glucose. So it would be turned on all the time because the cells are always converting glucose into energy. So it would need a steady expression of the gene so that there's plenty of protein for glycolysis. Another constitutive gene might be ones required for other processes in the cell that are called housekeeping. Okay, whatever, just for the basic metabolic process to keep a cell alive. So these genes would always be on. Okay, and in our experiment, the constitutively expressed gene will be the ampicillin resistance gene, because we have to have the gene on all the time so that the cell is resistant to the antibiotic that will be in the plate. The other class of genes are conditionally expressed. Conditional expression. Okay, and conditional expression means just what it says, that a condition is required. It requires a condition to be met to turn on the gene. Okay? So that's conditional gene expression. And it could be either tissue type, so where in the body or where in the plant the cell is located, or it could be due to hormonal response. So estrogen and testosterone are small molecules that affect gene expression. They turn on genes that are required for the reproduction of the species, especially in animals. It could be a small molecule. So organisms like E. coli prefer to utilize glucose for glycolysis, but it can use other sugars, and it has the genes to utilize those sugars. But generally those genes are turned off because it would prefer to use glucose. And so there's no reason to make those proteins if you're not going to utilize the sugar. So, but if the sugar is present, then the E. coli will turn on those genes. Let's say it's lactose. So it'll turn on the lactose genes so that it can utilize lactose in glycolysis. And so that, in that case, the lactose itself is the condition. Lactose present in the media will turn on the genes required to utilize the sugar. Okay, so how does that happen? So we've talked about genes and we've drawn them with boxes like this. Okay, so here's our gene. Here's our start. Here's our stop. And then we have the genomic DNA that the gene is found in. So here's our big long chromosome. Here's the gene itself. And then here is the DNA that's between the genes. So there'd be another gene out here and another gene down here. Okay, so this is called intergenic DNA. Okay, and here's our exons and here's our introns. Upstream of the gene in the intergenic DNA is something called a promoter. Okay, and the promoter is where RNA polymerase binds to. Okay, so we'll draw RNA polymerase with as a red triangle. Okay, so this is RNA polymerase. Okay, and so the RNA polymerase binds to the DNA sequence here, and then it starts transcription. So the promoter is a DNA sequence. Okay, and proteins such as the polymerase bind to the promoter and then can start transcription. So in a constitutively expressed gene, such as glucose 6-phosphatase, the promoter is always accessible. The RNA polymerase is always binding to the promoter and starting transcription. And then, of course, from that you get translation, and then a protein is produced. Okay, and that's constitutive. Nothing ever turns this gene off. Now then, conditional expression, gene expression. Okay. Okay, so in conditional gene expression, what happens is the okay, so here's our gene again, a new gene. And then this is a stop codon. Okay, and here we have our promoter. Okay, but in this case, if the gene is turned off, because there is a transcription factor there's a transcription factor that binds the promoter, and that prevents RNA polymerase from binding to it. So that can't bind here to the promoter because the transcription factor is in the way. Okay, but if a small molecule comes along, like a sugar or a hormone, okay, and it binds to this, okay, what happens is the transcription factor comes off of the gene because it's bound to the small molecule. Then the promoter is opened up, and now we're at like this. So that results in, and then we have the promoter, and now that allows our polymerase to come in, bind, and start transcription. Okay, so again, in the case when there's no sugar, the transcription factor binds to the promoter and prevents RNA polymerase from binding to the promoter. But if a sugar is added, in this case, okay, then the transcription factor binds to the sugar, releases the promoter, and the polymerase can bind to the promoter and start transcription. So the condition is the sugar, or if this was in a human and this was estrogen, estrogen would bind, release the factors, and then that gene would be transcribed and for some part of the reproductive cycle. So that's conditional gene expression. And like I said, it could be a small molecule like a sugar, a hormone, such as the testosterone or estrogen, or it could be positioned within a cell. So two cells touching each other can send signals that alter gene expression through the regulation of these transcription factors. Okay, so for experiment 3, we're going to be looking at the expression of two different genes. Okay, so on the plasmid that you're going to be given, okay, so a plasmid is a small circular DNA. Okay, so plasmids are small circular DNA pieces, and this one that we use is called pGLO. Okay, and P stands for plasmid, and GLO is the name, so pGLO. And pGlow has two genes on it. It has the ampicillin resistance gene, okay, and it has the GFP gene. Okay, so these are the two genes on this plasmid that are relevant to our experiment 3. So ampicillin resistance will confer resistance to the antibiotic ampicillin. It will destroy the ampicillin so that the E. coli can grow on the plate. The GFP gene, when expressed, will be transcribed and then translated into a protein that will result in green fluorescence, or the cells will glow green. Each of these has a promoter. GFP has a promoter upstream of it, and ampicillin has a promoter also. Okay. Now the ampicillin promoter is constitutive. We need the ampicillin expressed at all times in order for these cells to be able to survive on the media. So ampicillin gets a constitutive promoter. GFP, on the other hand, though, is conditional. Okay, we've designed it so that it is a conditional promoter. So and the condition is going to be a sugar. Okay, what sugar you're going to discover. So the signal is going to be a sugar, or the condition, in the media that's on the plate that the cells will be growing on. Okay, and if the proper sugar is present, then you'll get transcription. So this is if condition is met. Okay, this gene will get transcribed into an mRNA. Okay, and remember, this is going to be in a bacteria, so there's no splicing. We go transcription, and then we go directly to translation. Okay, and so that's going to give us a protein called GFP, for green fluorescence protein. And when the cells are exposed to UV light, okay, this protein will absorb the ultraviolet photons and will give off a green photon. Okay, so absorbs high energy ultraviolet light and gives off a lower energy green photon, and the cells fluoresce green. Fluoresce, okay? So they fluoresce green. So that's how you know, that's the report. If you see this green fluorescence, report that the promoter is actually turned on and being expressed. Okay, so this is a reporter for expression. So how are we going to do this? So we got this plasmid in one tube. Okay, we bought it from a company, and we have our E. coli. So we need to get the two together so that we can get the plasmid inside the cells and look at expression. And this is called the process of transformation. Okay, so anytime a scientist takes DNA from another organism and places it into a different organism, that's called transformation. The organism we're going to be working with today is E. coli. Okay, it's a common bacteria used in the lab for this type of experiment. But E. coli doesn't naturally take up DNA. It needs to be made competent. So in order to take up DNA, cells must be treated in ways that make them competent. If the cells are competent and we mix them with this and we treat them in certain ways, then they will take up the DNA. So the process is very simple and is demonstrated for you by Dr. Katie Burnett in the video that we made in the lab to demonstrate these steps. So the first step is to mix the competent E. coli with pGlow. Okay? And in your experiment, each student is going to use a different volume of DNA. That means a different amount of DNA, so that we can also look at the mechanics or the mechanics of the transformation itself, in addition to studying the regulation of gene expression. So then we place on ice for 10 minutes. Okay, and this just simply gives the E. coli time for the plasmid to stick to it, so that the plasmid and the E. coli get together and stick to each other. And we keep them on ice because we don't want the cells to start dividing. If they divide, they're no longer competent. So everything is done on the cold until we're ready for the cells to divide. The next step is to heat shock the cells at 42 degrees Celsius for 30 seconds. Okay, so we heat shock at 42 degrees for 30 seconds, and what's that is happening is that the membrane melts and the cell wall melts just a little bit so that the pGlow can enter into the cell. So it's during this step that the pGlow is going into the cell. We briefly return to the ice just to cool these cells down quickly, before we add a liquid media called SOC. Okay, and this is a really rich media. It has sucrose and other compounds that let the cells recover from this harsh treatment that they've just received and also begin dividing. So then we're going to shake them at 37 degrees Celsius for 1 hour. Okay. And we shake them to keep the liquid aerated so that these cells have oxygen, because they need oxygen to use the sucrose that's in the liquid media. The next step is to plate the bacteria. Okay, and there are 5 different plates. And the video will go through those different plates, and we will discuss it in class as well. So the 5 different plates. And then lastly, you incubate at 37 degrees Celsius for overnight, which is 10 to 15 hours. Okay, so this is an overnight time frame, which is 10 to 15 hours. And at the end of the incubation, which is generally the next day, if your transformation was successful, you will see on your petri dish a bunch of colonies. Okay, and each one of these colonies represents one cell that landed at that place and then divided overnight, thousands of times, so that we result in a visible colony of a clone of cells. And we can count these, and we record those numbers in our notebook, because we're going to use those numbers to look at not only we're going to talk about transformation efficiency, but also how well what does the effect of the amount of DNA have on transformation. Okay, so there's going to be a couple of questions that we're going to discuss. So we're going to record those numbers, and if the numbers are too high, then if it's about 150 or less, you just count everything and record that number. If it looks like it's way too high, you can divide the plate into quadrants and then count just one quadrant. So 1, 2, 3, 4, 5, 6, 7. And in your notebook, you would require 7 colonies times 4 quadrants equals 28 colonies. Okay. So that's what you will record in your notebook in this way, so that you know that this number is an estimate. The next step, what you'll do is you'll take each of the plates that have the small sugars, arabinose, galactose, or lactose, and you'll place them under ultraviolet light. Okay. And if the condition is met, and we get the gene turned on for GFP, we should see green fluorescence. So these cells will glow green when we expose to UV light. Okay, so that's how you'll know if the condition was met, because the fluorescence will report gene expression. Okay, so that's experiment 3. We will discuss in class some details about the different parts of the experiment, but this is an overview to get you started.